So it's time to go forward with our next event, and that is a panel, The Migration of Life in the Universe. And of course, this is continuing on the theme that we set with our opening session, that is, what are the natural features that go into setting the stage for natural panspermia, lithopanspermia, or if you can think of any other panspermia that's natural, feel free to trot it out. So we've invited some wonderful colleagues here. Lindy and I worked very hard to get folks to come at near the end of a semester, right? So those of you who teach have had to take time out from your busy schedules to join us. So we have Bill Bottke sitting next to me, David Catling, Ben Clark, Lindy, once again, Karen Meech, and Jay Melosh. And we're hoping that by having all these folks rub their clever brain cells together, that we can actually turn up some overlapping questions, some overlapping interests, some overlapping concerns, and perhaps some overlapping directions for research in everybody's individual areas. So what I've asked everybody to do is spend 3 or 4 minutes just introducing themselves very briefly beyond the introduction I just gave, and then giving us their main punchlines with respect to what they've been thinking about for this panel. So Bill, I'm going to start with you.
Okay, and you want us to start with a bio?
Yeah, a very brief bio.
Okay.
You were born.
I was born.
Okay.
So, all right, so my name is Bill Bottke. I'm the director of the Department of Space Studies at the Southwest Research Institute in Boulder, Colorado. I'm also the director of one of NASA's Survey Institutes, which is studying the places that humans may go in the future, basically small asteroids, the moon, and Phobos and Deimos. Okay. So my background is solar system dynamics and early bombardment. So I'm interested in things like what happened to the moon and Earth very early on in solar system history in terms of bombardment. How did planet formation take place? The solar system instabilities that we described earlier, and where planet migration is taking place and moving objects around. This is also something I worry about. And then I'm also concerned with asteroid evolution, meteorites, and so it goes on and on. So one of the really interesting things I think that I've seen at the meeting, and I think this is sort of backs up some of the things in my research, is that all of our current knowledge of planet formation, our current knowledge of how big impacts happen on worlds like the Earth and Venus and the rest, and also just our sense of dynamics of how small bodies move, all these put together suggest that there is communication, for the most part, between the different worlds of our solar system. You know, in the sense, we sometimes say around the departments in a colloquial way, you know, the different worlds all swap spit with one another, right? This is what happens. So we've seen this from observational evidence, from lunar meteorites coming to the Earth, from Martian meteorites. Dynamical models suggest that material from the Earth has the opportunity to not only reach all the terrestrial planets, but also to go out to icy worlds like Europa and Enceladus, which may have subsurface oceans, and they also go to Titan. And now and I think Karen will say much more about this, we've now recently seen evidence for interstellar objects that come in. Okay, and what they suggest is that if planet formation processes there are similar to what they are here, and that you're ejecting, let's say, 10 to the 15th, let's say Oumuamua size objects, maybe roughly, you know, somewhere between 100 meters and a kilometer or so, some of that material will have the opportunity to reach other systems, and that's what's happening in our system today. We think that something like an Oumuamua is maybe, there's always one within about 1 AU of the sun at any given time, and that actually has some really interesting implications. So one of the big issues then is not so much dynamical communication, because I think that can happen, but the issue is can material, let's say material with life or something interesting, can that life survive the passage it takes going from one place to another? Can it survive the impact velocities you experience when you may hit the Earth and the rest? And then even if you get there, okay, does that material have any, not to use a pun, but have any impact on the evolution of life in that world? And let me just give you a quick example. So for interstellar objects, something that everyone should sort of keep in the back of their minds is that anything entering our system gets accelerated by the sun, right? So by the time you reach the distance of Earth, 1 AU, the sun has already accelerated you to 40 kilometers per second. Okay, so the odds of you hitting the Earth at very high velocity is going to be very, very high. There are ways around that. You can capture objects, you can do various things, but generally most things coming from outside our solar system are going to be high velocity impacts, and so life has to be able to sustain that. In terms of other communication between the worlds, as we saw this morning, Martian meteorites can come here. A question that would be very interesting to ask is if we want to say Mars life is really the ultimate origin of Earth life, why is that so? What made conditions for life more conducive there than here? Is there some rationale that we can come up with that would say that's a more likely way to proceed rather than just saying that all life on Earth is local in derivation or so? So I think those are some of the main questions I have, and I'm actually very glad to be at this conference, and I'm very interested to see how people proceed and give us answers to some of these questions.
Great, thanks so much, Bill. Dave, weigh in for us.
Sure. So I'm David Catling. I'm a professor at the University of Washington in Earth and Space Sciences and also the cross-campus astrobiology program. My interests are, in particular, how atmospheres evolve on planets and how that can enable life or frustrate life, and how life affects atmospheres. So my particular interest in this is how panspermia kind of cross-cuts quite a few issues of astrobiology. So earlier we heard from Natalie Battaglia, who talked about pillars of astrobiology. And I guess I would have slightly different pillars. I see the kind of 4 pillars as being searching for life in the solar system, searching for life on exoplanets, SETI, which I put in a different category, and then also there's the origin of evolution of life on Earth, which is our one example. And panspermia essentially cuts across all of these issues in one way or another. But there is, I think, my perspective would be that there is an elephant in the room or an elephant on that list, and that is the origin of life, because no matter what the dynamics tell you, if your Oumuamua don't actually have anything inside them, there is no life there, life didn't originate if it's a vanishingly small probability, then you simply won't do the panspermia. Where does nobody said this, but the word panspermia is Greek for all seeds. And you're not going to scatter many seeds if you don't have any in your hand. So it seems to me that solving the origin of life, it should go hand in hand with this issue of whether panspermia is possible or not, because it's just a factor that can perhaps dominate if it's a very small multiplication factor for the probability. So that got me thinking, well, can we test something to do with the origin of life other than the kind of things that Steve Benner is going to tell us about this afternoon when you play around with organic chemicals in the lab? Can you test something in the solar system? And I think when we look at Mars, we don't see obvious signs of life that we do in the geologic record of the Earth. So for example, I've traipsed around in Northwest Australia with my colleague Roger Buick, looking at some of the most ancient signs of life on Earth, and they're kind of obvious in some ways. You see stromatolites, you see black shales, so fine-grained sediments that are black by virtue of how much organic carbon there is. These have never been discovered on Mars, black shales or stromatolites. So maybe it just didn't get that far on Mars if life began. In other words, it didn't get to photosynthetic life, which is doing that kind of thing. But maybe it didn't even get as far as very simple life. And in fact, maybe Mars holds the answers to the origin of life. And so I think one perspective that hasn't been advanced very much is that when we get samples back from Mars or we go to Mars, we should bear in mind that there may be some prebiotic chemicals, and maybe we could partly solve the origin of life problem by investigating things on Mars. So for example, I think Steve will tell you how cyanide or chemicals derived from cyanide can be extremely useful in making prebiotic chemicals, because basically it donates a pair of electrons, and it's a way to attack a carbon atom and add another carbon atom and over again and build up big molecules. So if you have cyanides and ferrocyan, you make ferrocyanides, which are relatively stable minerals. So we should look for those on Mars and make sure we go to the right places. And we should try and look for the simple 3-carbon or 4-carbon species that occur in prebiotic schemes and see if they're there on Mars and see if there are signs of anoxic environments on early Mars. It's oxidizing today, but in the past, we think life would have originated in an anoxic environment. So I think there are things we can do in the solar system that maybe can inform this factor that then will bias whether panspermia is possible. And I think that has to feed into the arguments, along with the things that Bill just talked about the celestial mechanics.
Great, thanks, Dave. So Ben Clark, go for it.
Okay, thank you. So I'm Ben Clark. I'm semi-retired. I work through SSI currently. My interest has been primarily Mars throughout my career, so I've been involved. I had an experiment on Viking, and then I've been involved with Spirit, Opportunity, Curiosity, and the upcoming Mars 2020 rover, various experiments. I've also recently become more convinced that Mars, if any place, should have been an origin of life as much as Earth, and maybe more so. Steve Benner kind of first pointed this out, and some of the discoveries we've made for geochemical elements that could be catalytic to life, plus the water-rock ratio, which gives more favorable opportunities for wet-dry cycles, makes Mars a primary place for the origin of life. And so we've kind of gone full circle from Viking, where after Viking, everybody was giving up on Mars for many, many years, 20 years to be exact, before we had another mission, until rovers opened up the variety, what I call the geochemical zoo that we're discovering on Mars. I also work on comets and asteroids, and I think there may well be an important part to play for those objects in the origin of life. But I'm particularly happy to be back in Berkeley. I was actually out here as a little boy at the tail end of World War II, while my dad was sailing around the world at courtesy of the U.S. Navy. And then about 15 years later, I was a graduate student here in nuclear physics and engineering. And then later, I kind of got religion, so to speak, and went into biophysics, which was a new discipline, and I went back to Columbia for that. But another event that happened here in Berkeley was the 8th meeting of ISIL, the International Society for the Study of the Origin of Life. They only meet every 3 years. And that was in 1986. And at that meeting, there was 2 abstracts. I did one, and the other was Gerda Horneck, the German astrobiologist. And our topic wasn't important, so we were relegated to posters. But that's the way we got to know each other, because we had posters right next to each other, and they were on the same topic. Mine was called Barriers to the Interchange of Material from Earth, Biologic Material from Earth to Mars, and hers is similar. So I was very worried about what little scientific career or credentials I had being essentially torpedoed by even talking about something like this. And I don't think I ever used the word panspermia on purpose. And Gerda was also kind of looking at the same way I was looking at that. Namely, it seemed possible, but there were all these hurdles, like the shock of the departure. Everybody thought at that time still that to get a meteorite off of Mars, you had to melt it. And Jay solved that problem, so you'll hear more about that. But I strongly believe that it might be possible, although there's one thing I think that is relevant to Mars that maybe hasn't been given enough consideration, might make it a little harder than we think, and similarly one thing at Earth that might also make the probabilities a little lower, but nonetheless, you have to multiply several probabilities together. It's not high, but nonetheless, there's been a lot of impacts and a lot of interchange of material. So maybe life did come from Mars to Earth, and maybe it was easier to start on Mars. And I like David's suggestion that maybe if we really find a good handful of organics on Mars eventually, it may give us important clues as what prebiotic chemistry, how it proceeds on the way to the origin of life.
Thanks, Ben. Lindy, your thoughts?
Lindy Elkins-Tanton. I'm the principal investigator of the NASA Psyche mission, and at Arizona State University, President Michael Crow and I co-chair our interplanetary initiative. And although most of my research is about how you build terrestrial planets, and the temporal and spatial nature of habitability comes and goes at different depths, different times, Now what I'm spending almost all my time thinking about is how we can use the motivation and the inspiration of sending humans and robots out into space to actually cause our human society to move faster in terms of learning how to solve problems better and be a society of problem solvers who are better educated with better agency. That's what I'm mainly thinking about now, the human panspermia and the ways that we can use that to motivate to make a better society.
Great, thanks. Kara?
I'm Karen Meech. I'm an astronomer at the University of Hawaii's Institute for Astronomy. I began my career specializing in comets, and now this has migrated more to using small bodies to understand how habitable worlds form. And of course, lately, with the advent of all the new big surveys, I've been spending a lot of time following up interesting discoveries from pan-stars. And so to that effect, we all know that we've had our first discovery of an interstellar object moving through the solar system. And this is very likely a cometary planetesimal that escaped from its solar system at some point. And we know that it's cometary because it experienced non-gravitational acceleration. It was accelerating as it moved out of our solar system, much like comets in our solar system do when they approach the sun. So this tells us that ices can survive over these interstellar distances. And of course, we knew that from some of the small body missions. The Deep Impact mission, the Rosetta mission, showed us that comets are low density, very highly porous, excellent thermal insulators, and you can preserve material just meters below the surface at very low temperatures. Moreover, there's been a lot of work, even back into the 1980s. I remember Alan Stern, and others had worked on what is the radiation like and how deeply can it penetrate into these small bodies. And you're probably getting significant protection on the inside, and you're not getting any deeper with the radiation field or heating for supernovae below 10, 20 meters depth in a body. So the idea that you could bring some living organism in some state, it could be preserved in a cold deep freeze, so it would be no different than coming from our outer solar system. Now, how long ago might this have happened? In the case of Oumuamua, tracing back its trajectory, Corin Baylor-Jones found that there were 4 candidates for its home solar system, ranging in time between 1 and 6 million years ago, passing within an Oort cloud or 2 of those star systems. Unfortunately, the ejection velocities would have been a little bit too high, so it's probably not those solar systems. But tens of millions of years might be the travel time or longer for this thing. We saw earlier today that there's likely at any one moment one of these of the size of Oumuamua on the 100-meter scale that could be within the inner solar system. Now, some of my questions concern could an impact of something like this have survivable biologic material? Oumuamua came in at a velocity of 60 kilometers per second above the plane of the solar system, so that's a very high impact velocity. Of course, if it come in the plane of the solar system in a head-on collision, you can get velocities of 100 kilometers a second or more. So that's one of my questions. And then finally, given the beautiful images we've seen from ALMA and the fact that for many protoplanetary disks, we see that the gas clears out very quickly, and that's an era when it's really easy to eject material. Would there have been enough time for life processes to start in the few tens of millions of years?
Great, thanks, Karen. Jay?
Okay, my name's Jay Melosh. I'm a professor at Purdue, and you can read my biography. I've been interested in impacts for a long time. I first got started in this area in the 1980s when it was speculated that a group of odd meteorites, this Shergottites, Knocklights, and Chassigny, might have come from Mars, and worked on a mechanism by which they might be ejected, this spallation mechanism. Having done that, it occurred to me, knowing the conditions under which you could get a launch of very lightly shocked material at high speed, it occurred to me that maybe living organisms could tolerate this. Not macroscopic organisms. The accelerations are hundreds of thousands of G upon launch, so you and I wouldn't make it, but maybe our microbes might, our gut microbes or something. At any rate, at that time, it was 1988, I submitted this idea as a paper to Nature. Nature did publish it. They wouldn't publish it as a paper They published this correspondence, and they chose a gag title for it because they thought this was so outrageous.
What was it? What was the title?
The title they chose was The Rocky Road to Panspermia. Panspermia was not my word. It was the editors of Nature.
Rocky Road.
At any rate, from that start, I then got together with a biologist at Arizona, a guy named Wayne Nicholson, and he got very interested in this. We started subjecting bacteria to all of the things that would be required to get them from Mars to Earth. We accelerated them to hundreds of thousands of G. They liked it.
They liked it.
We shot them from guns, and at least one variety, Deinococcus radiodurans and archaea, actually, we had more after the shots than before, so they liked it very much. And we also did high-speed gun experiments, injecting blocks of our genetically labeled bacteria from blocks of rock, collecting the debris. We labeled granite discs and glued them on the outside of sounding rockets and let them re-enter and measured the biological content there. We collected rocks from various places around the Earth and measured the biological and initial condition of launch from the Earth. So we did a lot in this direction of trying to evaluate whether or not it's plausible that things might have been exchanged from Mars to Earth. And I'll remind you that about 1 ton of Martian material falls into Earth's atmosphere each year. So it's not as if we're looking at one rock every now and again. Only a few, now 100, Martian meteorites have been picked up. And by the way, Martian meteorites, unlike lunar meteorites, which are all finds, there are Martian meteorites falls, the most recent being the Los Angeles meteorite. So, you know, our connection with Mars is close. And furthermore, from the conditions of launch, what we get into space is all from the near surface. On Earth, that's the biologically most active area. Now on Mars, because of the radiation, maybe at least on modern Mars, that's depleted because of radiation near the surface. But even from tens of meters, and some Martian meteorites do not show evidence of pre-irradiation before launch. If there is stuff down there living at meters depth, there's a good possibility we might find it, or at least they would be able to make the trip. I've also tried to evaluate interstellar panspermia. Some of you may know my 2003 paper. I know some of you in the audience have done their best to shoot it down, but worked out what is the probability of a rock from the surface of a planet, like the Earth in our system, getting to another planet in another solar system. And I'm not talking about just, you know, something from our solar system, but something specifically from the surface of our planet. Now, of Earth ejecta, 30% is caught by Earth, maybe 10% goes to Mars. A higher percentage, about 15%, gets ejected entirely from the solar system by interaction with Jupiter. And the way into another solar system, rather than directly, is to be caught into a cometary orbit by a giant planet, which will then, well, on the next go-round, may either eject the thing entirely from the solar system, or it could be caught into an orbit like a Jupiter family comet, and would then have a much higher probability of encountering a planet. The encounter velocities would be, of course, lower. But even so, putting that all together, the probabilities and the very long exchange times, I think it's very unlikely that by natural means you could exchange material from one planet in a solar system to another planet in another solar system.
Great, thanks. So that's the menu that we have on offer. I've got a few questions that I want to direct to you guys, and then we'll open it up for general discussions, unless you take off on a different pathway, and that's fine with me. I didn't want to overstructure this. So one of the things that I think has come up in discussions on panspermia, and that is the Mars-Earth case is well considered, right? People have paid attention. We had a a presentation on it this morning. One of the things that's been pointed out as a feature that would be an asset for finding any kind of life processes on icy moons in the outer solar system is that it's more likely, or would definitely be, a second genesis of life. On the other hand, you know, from what I've heard everyone present in other discussions, it seems like there are plenty of transport mechanisms that could actually transport material far enough to actually intersect these outer solar system objects. What are the impediments then that we might have that would interfere with a successful potential transfer of life either from Earth to one of these other bodies or from one of these other bodies here? And I'm not going to call on people. I would like you to respond if you have some insight into that question.
Well, I have
Yeah, go ahead,
a
go
quick
for it.
answer.
Yeah.
And that is, if it's not impossible that Earth material can go out and encounter the moons of the outer planets. It's something that we've calculated, and it probably is low, but it's not zero. The problem with a rock from Earth making it, say, to the oceans of Europa or Enceladus is that they don't have atmospheres, and the soft catch potential of the Earth's atmosphere isn't present there. So you're going to have a hard landing at high velocity, and it's not terribly probable that living organisms would survive that. On the other hand, going the other direction, if you could eject ice from the surface and get it to Earth before it evaporated, you might have some chance there. So that's my 10 cents.
I'll mention the one exception to that would possibly be Titan. Provided Titan's atmosphere was thick in the deep past as it is today, you can imagine some kind of aerobraking for material there. It's still, the odds are very low, but it's not zero. So that might be a possibility. Well, one thing I think I want to mention about in terms of your other case, about what are the impediments.
Yeah.
Something to think about is that, you know, our solar system is growing planetesimals at its earliest stage, right? So it's making objects similar to comets, making objects similar to primitive asteroids and the rest. And much of that material is being ejected. Only some of it goes into making the planets. And other star systems are doing the very same thing, okay? So the question is, for the case of that Karen was talking about, like Oumuamua, that's very likely some form of planetesimal from another system. And so it might actually be, you know, remarkably interesting from a science perspective, but does that material have is it fundamentally different than the planetesimals we're growing in our solar system? I mean, to really transfer life from another star system, you probably need to get ejecta from some kind of terrestrial planet. And then you're not talking about kilometer-sized objects. In most cases, you're talking about much smaller objects, and can they make the passage with everything surviving? And I think that's an important distinction that we have to have when thinking about panspermia within the solar system and those in going interstellar distances.
Of course, one of the questions with Oumuamua that's still very outstanding is the explanation for its strange shape. And there's been a lot of creative speculation that while it could be just a regular planetesimal, maybe it was due to some sort of tidal stretching or some sort of other process, and it could have been part of something much bigger at one point.
So
I would
planet
just-
shatter.
I would just add one obstacle. So,
Yeah.
so Bill mentioned Titan, but of course, if Earth life went there today, it's not going to do very well at 90 Kelvin and with no liquid water. But there is a possibility. There have been papers published some years ago by Jonathan Looney about the early atmosphere of Titan, possibly making it warm enough on the surface, and so maybe we would have to think of a transfer very early in the solar system to Titan to make that panspermic, if you like.
Or maybe it's waiting for us now, Dave. Right? I mean,
Well, if it's liquid water-based life, it would have to be deep under the surface, or it would just be dead.
no, I mean, you know, if bits of life from elsewhere were transferred to Titan in its present chilly condition, it would be like, you know, lyophilizing bacteria, which is the way we essentially store them here.
Yeah.
So one might then say there might be the potential for a nascent biosphere on Titan until our sun goes into its red giant phase, and then that shifts the habitable zone out far enough to actually wake those guys up. So maybe we should be looking for them at some point in the future.
And let me just say this: if we're going to talk about early Titan, then I think we also get to talk about early Venus, which is Venus is like the
Yes,
very
Venus.
ignored planet, which is unfortunate because it's really a
Very
fascinating world, right?
unfortunate,
But Venus early on, for all we know, had as much water as the Earth did. There's reasons why Venus would lose its water over time. Venus today is a very unhospitable place, but it could be early on, it wasn't very different from the early Earth. And transfer between those two planets could be very interesting. So I think we shouldn't neglect Venus in this whole story, even though today it's very hard to think about getting samples from it and the rest. But it may have been important in this story.
Well, if we're going to have early Venus, we're going to have early Triton too.
Yeah, okay.
Because when Triton was captured for Neptune, I mean, those models show that it probably had a liquid surface at that point, and so that would have been a good target for panspermia.
And if we're talking about early things, I think that I would like to say, especially to this moon-loving audience, that the earliest rocks from the Earth are no longer on the Earth; they're on the Moon.
Right.
We've had a complete resurfacing,
Yes.
and we got to go to the Moon and find out if we can get the date of the advent of Earth life back further.
Yeah, and maybe even preserved organics on the inside of those objects. So, yes,
So if
Ben?
Venus was hospitable early on, maybe Earth seeded it. And it
or
didn't-
vice versa.
didn't survive.
Or I think it's a fascinating question. I think we tend to ignore Venus in all these conversations, but I don't think we should. But I think it's hard to test these questions. That's the problem.
Well, I was going to say that too. It's going to be very difficult to do much about Venus because it's so hard to do missions there.
Yeah.
Now,
Exactly.
relevant to other things that are hard, one thing we don't know very well is how well can organisms withstand ultra-high vacuum. We know that some do degrade over measurable time intervals, meaning months or years, but you can't do billion-year experiments in the laboratory. You can try to speed up the process with temperature or something, but there seems to be evidence that ultra-high vacuum can, in fact, slowly damage bacteria, which might make it more difficult to go interstellar, because there you're talking about very long time periods.
Is it the vacuum or is it the cross-section for cosmic rays that is the real issue?
Well, no, actually, spores survive radiation better in vacuum than they do wet. So,
because of lack of
so
oxidation.
it's probably, I mean, one factor is just the loss of water, which stabilizes the chemistry of the cell to some extent. And so the proteins essentially are becoming, inoperative. They're losing, because they use hydrophobic bonding for some of the structure. So it just might be difficult, but it's again going to be difficult to prove how long spores can survive under these conditions.
Yeah, mostly it's conjecture.
Conjecture. Ideally, you may want them to be in an icy body that stays icy, because once you're away from a solar system, you're going to be very cold.
Yeah, and protected by that ice layer. Jay, you had a comment?
Well, I might make the comment, working with Wayne Nicholson, we tried to understand how it is that spores could survive the millions of years in space needed for a most likely transfer. And what we found, I mean, he started out as a food chemist trying to actually kill bacteria and the mechanisms by which they survived the various
Send them to
insults
space.
the food industry gives.
Then eat them.
And radiation is one of them.
Yeah.
Yep, that's one way of preserving food. What we found, or he found, is that when the spores are formed by Bacillus subtilis in particular, it wraps its DNA in a histone, a short piece of protein that actually preserves it. I mean, normal DNA, unprotected, degrades thermally reasonably fast, and you wouldn't expect DNA to last, millions of years. But this wrapped DNA actually will survive, and that's the intent of the spore, is to stabilize it. Possibly for millions of years. There are reports of bacteria surviving 215 million years. They're, of course, highly controversial. But evidently, this process of wrapping your DNA is something that helps preserve things. One of the things we did years ago with a graduate student named Rachel Mastrapa, who, I don't know, she might be here or in the audience, she had a project in a genomics class, and so we knew the DNA sequence of this histone, and so she went looking for that and asking where in the tree of life might we find this protein sequence that preserves us against vacuum. Our life has a very great tolerance for vacuum. And why is that? Well, what she found is that the genome sequence is in every branch of the tree of life. It's in everything. It's in us right now. It's doing different things, but it's everywhere. And so one could wonder then, well, why is it that all life on Earth has buried in this genome a sequence that protects its DNA from vacuum and radiation? Why did we need that? And I'll leave you to answer that.
That's awesome.
Pregnant
That's
question,
awesome.
Hank.
That's a mic drop moment. We can all just go home now, I think.
So if you had to make a decision about what, in your particular area of interest vis-a-vis panspermia, had the biggest question, had the potential for a showstopper, about which we know nothing, but you think that there would be a potential to do work to actually make progress on that, what would you pick?
Well, I have one that I alluded to. I think one of the concerns is that the Martian meteorites are all strong igneous rocks. And so the Spall mechanism probably works very well with strong rocks, but what about weak sediments? And on Mars, we're finding everything is weak. Even the things that analyze as being igneous are not as hard to abrade or drill as most terrestrial igneous rocks. So where organisms are most likely to be near the surface anyway is in some kind of sediments or weathered material. So that's something that could be studied, I think, to study the strength of the Martian meteorites as we know them, to try and get a lower limit. And then Jay's work, maybe with experimental work or even theoretical, could help establish that limit.
So the source depth is what you're talking about?
Sorry?
The source depth?
Well, it's just the strength of the sediment that the organisms are in. If it's a really strong material like a chert, they're probably no longer biologically active, because now they don't have the porosity and the access to liquid water.
But that's a really good place to preserve dead stuff.
Exactly, exactly.
Because that's what we get
Yeah.
from the Archean Earth, is chert is a good material.
Right, exactly. But for panspermia, we want the opposite.
Yeah, in fact, that observation is entirely consistent with what we see in the Martian meteorites. The Martian meteorites do not contain sedimentary rocks, except possibly the hydro-cemented 8401. Most Martian meteorites are igneous rocks. They're strong, and yet there are large areas of Mars that are covered with weaker rocks. Probably they don't make it off the surface as big rocks, and that's the reason that we don't see them in our collection.
Or at least they don't last long enough to
Karen, you
enter
have one, right?
Earth's
Well,
atmosphere.
and sedimentary materials that land on Earth may not be as easy to find as hard igneous rocks that have a nice ablation.
That's right. Would we know them if we saw them?
Yeah, exactly.
Well,
I would think
bunch of
you'd
them
have
are
a melt
false.
crust still, right? You have the same thermal effects coming in, so you presumably would produce some sort of a rind. I wonder if that we're just not
Well,
recognizing.
but if they're soft and friable, that rind may go away a lot faster than it
Yeah.
does on a
Yeah, good point.
I'd like input from the biologists, because I still think the big elephant is it's easiest to eject the planetesimals very, very early in the solar system
Yeah.
building process. So how do you get life going that quickly?
Yes. Yes, you do.
Well, I think that's an article of faith amongst biologists, that you either do or don't believe that it's possible. And I'm not sure that, any of our opinions are terribly well-founded at this point.
Then that sounds like a good area where you really do
Exactly.
need the research.
Exactly. But it's a very hard area to work on.
Are there limits on how quickly it could happen?
I belong to the it's easy as pie to make life school of thought, because we have planet-size laboratories conducting zillions and jillions of simultaneous experiments in a plethora of different intersections of conditions. So I think that it's very hard for us as humans, even scientifically trained humans who use math as a proxy for reality, to imagine the power of that much innovation going on over a few million years. It seems to us like this has to be some, rare event, but in my mind, it's, a concatenation of all these experiments going on with gradually selectivity emerging out of that. Some chemistry is going to be more successful than others. But there are people who are very much on the opposite end of that spectrum. And that's a measure of our ignorance, Karen. I think you're exactly right.
Yeah, and along those lines, if exogenous material from comets or carbonaceous meteorites played an important role in the origin of life, as some of us believe it might well have, then that would be the ideal time. It's when things are all being mixed up and we're delivering all this material. So as long as you don't send two objects right in a row at the same spot, which is vanishingly small probability, then it may be favorable to have this material coming in.
Although I think there's I'm sorry, do you want to go ahead?
No, go ahead.
This is something we were talking about, Bill and I were talking about at dinner last night, is that there are two competitions around size. If you want an easy landing with something with an atmosphere, you have to have something that's large enough that as it's transiting through space, you can allow the outer, however many meters you choose, to be completely wrecked by high energy cosmic rays. So you've got the inner part that's maybe less irradiated, depending on how much irradiation you think your wrapped DNA can handle. And then as it comes through the atmosphere, it's got to be small enough that it's not going to explode with the power of its own shock wave and cause itself to become a plasma. And so you've got to have something that's small enough to send part of the interior part intact to the surface, but big enough to be shielded during space. And so that's a problem with planetesimals.
But let me try to ask maybe a question and a comment on this. Is that because maybe I'm missing the issue here? Because in planet formation, you're making planetesimals. You're making a lot of things, right? But I guess I don't think about a planetesimal as an abode for life. Although maybe that's the wrong way to think about it. There might be some people in the audience would say, hey, yeah, we could imagine, a 100-kilometer body having life. But
didn't like my talk this morning, apparently. Go ahead.
I'll have to start watching your talks again. No, but the issue I could see though is that at least the way I think about this is that, so you're during planet formation, you're making planetary embryos. They may be differentiating early. Maybe you have early water. Maybe you have the conditions, even for a very brief time, to have life. And then they're being bashed by other planetary embryos, other large objects, and you're creating fragments, and then they escape the system. So isn't that the main source of panspermia? If it is, then things are very early, but it's really those embryos that are smashing one another that really provides the greatest possibility of getting interesting things from one system to another, rather than, let's say, just scattering a bunch of comet-like objects across the interstellar spaces.
Well, the problem with that is that, we know for sure, as was mentioned this morning, that there are lots of organic molecules that are synthesized in space, but it's basically a tar. It's not biochemicals, and the biochemistry is very modular. So if you look at lipids, for example, they're made up of acetyl groups, which are 2 carbon groups joined together, or they're 5 carbon isoprenes. That's it. It's a Lego principle. And so you can see that these are not biological, the ones in the meteorites, because they don't operate on this Lego principle. And so I think there's still a lot of empirical stuff that can be done in the solar system exploration. Sure, let's fly through the plumes of Enceladus and let's check whether the lipids conform to the Lego principle. Let's check whether there's homochirality. If not, it's just organic stuff in the solar system, but not alive. But it could be alive. Well, let's go to Mars and check whether it got to RNA world, in which case you're going to find uracil, which is a characteristic base of that world. And if it didn't get that far, you won't find it. Or if it got further, you'll find something more complex. But I think, the way that we solve this problem is not by having an opinionated preference, but actually having data.
Yeah, that I agree.
But you don't get the opportunity to get data unless you have an opinionated preference.
Well, the opinionated preference is part of the motivation for
That's right.
flying through the plume of Enceladus
For
or
making
getting
sure
a sample return
missions
from
happen.
Mars.
That's
But,
right.
Yeah.
Yeah. Well, I think that we have taken up our panel time, and now it's time to open up to the floor. And so if you can raise your hand, as we've been doing, we're going to be throwing these delightful and ever amusing cubes at you. Or
Sell it, Dr.
the what?
Sell it.
They're blue, which is a lovely color, and he'll toss it to, right? Try not to put anybody's eye out or knock their glasses out. You can see that I love these things. And you can address it to any member of the panel.
Go ahead.
But you can also ask questions of the speakers in the presentation session this morning, if you had one that was a burning question that you didn't have a chance to ask.
Hello. Actually, a really good segue for what you guys talked about last, because my question is related. You guys talk a lot about, like kind of spores and fully blown organisms being blasted through space on small bodies or whatever, but I know that I've read that certain amino acids like glycine can form abiotically in space, and like what do you think about those maybe less complex but kind of starter biochemical molecules seeding planetary bodies as they form, as a way to like introduce biochemistry, not necessarily like a full-blown organism, but just kind of the ingredients to make that?
That's how they got here, I think.
Well, okay,
Yeah, go.
I would say that comets in particular may be exceptionally good for this. We don't know for sure yet, because we don't have good samples of comets yet until we do a comet sample return mission. But we have a lot of hints that it's different from the most carbonaceous rich, most organic rich carbonaceous chondrites, which do have quite a bit of organic materials of all sorts, but it's almost too much of a grab bag, and so that would have to be sort of weeded out to do biotic chemistry. But in particular, there's evidence that comets may have a higher nitrogen to carbon ratio. We had a direct measurement on stardust that seemed to indicate there's these things called UCAMs down in the Antarctic that have that. There's inclusions in meteorites that have the high nitrogen to carbon ratio, and on Rosetta they had unusual characteristics compared to meteorites. But this whole class of objects from carbonaceous chondrites, which meteorites which come, we think, from the asteroid belt, all the way out through the Kuiper belt objects and then even to the Oort cloud, may be really excellent carbonaceous material to do that. Now, that's all throughout our solar system, and there's every reason to believe that would be true in other solar systems as well. So you don't necessarily need to transfer from solar system to solar system, but at least along your thoughts, another object coming into a planetary like Mars or Earth may have seeded the beginnings of that synthesis that led to life.
Thank you. I don't want to throw it. Do we throw it?
So, should I go? The question I just wanted to bring up, when I was at the Planetary Society a few years ago, we tried to conduct an interplanetary transpermia experiment by creating an artificial rock, placing it inside the Phobos sample return capsule and sending it out to Phobos and having it return as part of the ill-fated Phobos sample return mission that the Russians were trying. So we're still looking for an opportunity to actually conduct a flight experiment of transpermia to go through the whole vacuum temperature cycle and the crash landing on Earth and 40 Gs of acceleration and see how they survive. So that's something in the future.
It's still on your bucket list, Lou. It's still on your bucket list.
Yes, it's.
Yeah.
Did your piece
We
of
still
rock,
have capsule ready to go looking for a sample return mission.
did your piece of rock have bacteria in it?
Yeah, we had 10 different microbes well, actually 9 different microbes and 1 soil sample that was a combination of organic material.
But to be fair, Gerda Hornick has explored, some important parts of that scenario by exposing microbes to open space. She had a number of samples on the very long duration exposure facility.
But that was inside the Earth's radiation field. It was inside the Earth's
It was inside the Earth's radiation belt, that's correct.
Yes.
Okay, I'd like to panel source the question. Powers of 10 only. No more accuracy needed. Everybody think of an answer. How much money should the human race spend to get a piece of Oumuamua? And that would include like planning it thorough material analysis. How many dollars? Sure, everybody has an answer, so let's, powers of 10.
Okay. Okay, I'm gonna, all right, so you want to sample. You don't just want to investigate the body, you actually want to sample back on Earth.
No, not sample back on Earth, but you want to know what it's made of, isotopic. Not how much would it cost, how much is it worth?
Okay, I'll give a ballpark number, and maybe this is useful. I would say if we could do that for the cost of a NASA flagship mission, I think that would be worthwhile. Is that a reasonable answer?
I don't want to tell you what I'm thinking of.
I agree. I'd say a flagship mission. And there are teams that are looking into this. There's been a number of papers that have shown that it is feasible, that you could have gotten to Oumuamua with launch getting there, when it's 100 AU from the sun, which makes it challenging. There are teams that are thinking about what to do for the second one that comes through, getting missions ready to go, parked in orbit so that you can meet them closer into Earth. So I think it would be well worth it at a flagship level.
It's actually not feasible because
Oh, you use the microphone.
Yeah.
can I just
Try it.
I'll return it. It's actually not very feasible to do for the simple reason that Oumuamua is on a trajectory that was not dictated just by the sun's gravity, so we don't really know exactly where it is. And in order to find it in the darkness of space, since the brightness drops off as 1/4 distance to the 4th power, in order to find it, you need to equip any such mission with a telescope that will search for it. And that's very heavy to carry, a telescope. You can't just have a gram-sized payload that will take a photograph of it. You need to send the whole telescope. And that becomes very expensive. The second thing is about these envisioned missions that would go close to the sun and get a boost, a gravity assist, in order to get a very high speed and chase Oumuamua, because chemical propulsion on its own cannot do the job. So there are these concepts where the spacecraft goes close to the sun. The only problem is that they have to come closer than the Parker experiment, the Parker mission went. And at that point, when you enter the vicinity of the sun, any material object will melt down, not to speak about the fact that it will behave like a comet because it will eject this gas from it that will deflect its orbit. So I would, irrespective of how much money you want to put into it, it's not worthwhile. It makes much more sense to search for the next interstellar object. And within a few years, we will have the Large Synoptic Survey Telescope that will detect, if these objects are on random trajectories, there would be once per month of a size of 100 meters detected by LSST. That's a simple projection from what the Pan-STARRS was sensitive to. And so just wait a few years and we'll have one per month. And then go after those with much less cost. If you detect them on their approach to us, you can actually meet them halfway at a relatively low speed. So.
Well, I think the LSST folks are predicting maybe one per year. But either way, it looks like we're going to have more than we can handle in the near future.
I think I would argue, too, that rather than try to do a sample return right off the bat, it would be much more cost-effective and help you decide if you could afford or wanted to afford sample return over other missions if you just got really good spectral measurements of an object like that and use both, visible to near IR and far IR. And you would be able to determine pretty quickly, is it like solar system material or is it something really different?
On that note, that's really not a problem.
Don't worry,
On
Andrew,
that
I'll
note, I just wanted to point out that if such an object passes close to the sun, and that's a paper that we've written with John Forbes, it will get evaporated. And you can calculate the statistics of that. And then if you just do spectroscopy of the gases that come off it, you can learn about the composition without the need to visit it or land on its surface. And that, such objects should exist out there, and we just need to monitor the cometary tail that comes off them when they get close to the sun. No need to send a mission that will land on their surface.
But someone might argue that getting the isotopes would be fantastic. So that would require sample return.
Yeah, here Jean-Pierre de Vere of the German Aerospace Center at the Institute of Planetary Research in Berlin. I was just to let you know about a comment to the vacuum, so the resistance of organisms to vacuum. I'm a PI of an experiment named Biomax on the ISS, where we have a lot of results where we have found that just the vacuum was not damaging so much compared to the radiation, because we had also simulated experiments, so Martian environment with very low density of atmosphere and as well complete vacuum. And we saw that especially vacuum seems much better preserve the spores and bacteria compared to the radiation, where there's some damage, and even we have now some hints on the DNA just for the bacteria and archaea that there is much more damage through radiation compared to vacuum. So this is just a comment that this is a response that vacuum seems not so depending on the species. We have also to mention this depends on the species. We have tested species, bacteria up to lichens and mosses, and the bacteria are much better resistant to these conditions compared to the much more complex life forms.
So I remember papers by Klaus Doze, who would have also been the same organization, I guess, showing survival curves as a function of exposure to vacuum. So there must be a variety of responses across different species.
So you could summarize by saying Earth life has an eerie tolerance for vacuum.
Yes.
Our microbial life is pretty good at handling vacuum. Lyophilization involves vacuum, and more organisms survive being microorganisms, survive being put in vacuum than almost any other treatment.
But to what extent is this adaptation just the same adaptation as dryness? So, people have pointed out that it's the same, essentially.
Yeah, it is the same mechanism that these organisms use for dehydration.
And if you look at simple eukaryotes, a lot of them have a cyst phase where they just sit around, and that can withstand like a dry spell or something, and then they'll come to life again. So I think it may be too much of a speculative extrapolation to argue that this is some kind of panspermia signature. It could just be a dryness signature because it gets dry on the Earth.
But on the other hand, if it's very common, which we know it is, then it means that a very large percentage of the Earth's microbiota would be a good candidate for actually tolerating those conditions. So there's also a positive side effect of that commonality. Andrew?
Yeah, I want to make a couple of comments. One of them is on this panspermia from Mars to Earth. There was a lot of focus on the transfer of material. And despite the fact that 1 ton per year sounds like a very unimpressive number to me, we have to, there is a number of other parameters that we have to count, probabilities, which is where this material comes from, what was the density of microorganisms on Mars, does it come from the area when were microorganisms or not, how much survived when it landed, how much survived when it after it landed. Remember that even, if you're a microbiologist, which I am not, but I worked enough with microbiologists, even a few degrees of centigrade could cause that bacteria don't want to replicate or replicate very poorly. So you have to have, and a kind of a broad statement, well, on Mars, early Mars conditions might have been similar to those on Earth, is not good enough. And here I fully agree with David, who said that origin of life and transfer of life are very, very close related topics, We not only want to know whether there was life on Mars, but what kind of life it was. And kind of vague statement that it could have been similar, conditions could have been similar on Earth is not good enough, because what decides eventually is the weakest link. Yes, that several things may be similar, but there may be one thing that is missing when it landed on when it landed on Earth that it cannot get, and it's not going to live. Or it could live very, very poorly, not to proliferate, and eventually get destroyed. So we are very, very far, but even saying how much material gets transferred, we get very, very far from arguing that panspermia is a feasible phenomenon. Now, regarding panspermia elsewhere, there was this talk about Venus and early Venus and early Titan, and I don't know whether there was water there or not, but even if there was water, and even if life got transferred there, it's not there today, because there is really not a shred of evidence, of serious evidence that life could be non-aqueous. So and probably any trace of it is long gone. So if we can't find it, any trace of it, why we even bother about talking about it? It's sheer speculation. And my final comment is kind of related to something that Jay said, and that was on histones, and he kind of posed this question that sounds good when you say it fast, which is, why all those histones are there that are similar that are similar to those that make it resistant to vacuum, and it kind of implies that vacuum was the first function of those histones. What is really a hallmark of proteins, of terrestrial proteins as we know it, is that many different proteins that perform many different functions have very similar structures. A great example is Tim-Barrel proteins, that 10% of all proteins with all families of functions have the same architecture. So there's nothing special about it, that you find it everywhere doing everything else. So it's not an argument for anything. Thanks.
Responses?
Well, to your first point, I agree that was my other factor that I think has been underappreciated, is that even if we transferred organisms from Mars, they would have to land in an area on Earth at a similar enough environment that they could go ahead and metabolize and reproduce. So, for example, if they were photoautotrophs, that might be easy. But on Mars, if you're a photoautotroph, you have to be able to handle the UV, and that's very difficult to do. So you either normally live away from the sun and use some kind of chemolithoautotrophy to survive. And the problem with that is then you need to land in an environment on Earth where the ingredients are available. So, for example, Mars is just loaded with sulfate. And currently the hydrogen concentration on Mars is a little too low to make that a viable metabolic reaction. But we know, or we think anyway, that it had a lot more hydrogen in the past, and it may also episodically get more hydrogen during impacts or volcanic activity. So there may be sulfate reducers on Mars sitting there. They come all the way to Earth, but they don't land in the right place on Earth, and they drown in the ocean. And then get eaten before they can reproduce.
Somebody had, okay.
So I have one question and then a comment, which is also a question. I would like to hear from you some consideration about the timing of the various histories which are bringing to our existence in this planet and the life. I mean, the Earth started 4.5 billion years ago, okay, approximately. It cooled down, and immediately after the prokaryotes started. It was very quick. As soon as the conditions were cold enough and stable enough, life was everywhere, and since there has evolved. Mm-hmm. So this is a very short time scale, because after the Earth has formed, after the conditions were stable enough, maybe there is an uncertainty of a few hundred million years, but this is a time scale. I would like to see this time scale of a few million years against the time planets talk together, the times we get Oumuamua coming in, the timing, all the various timing of formation of planetary systems, because this could help us understanding if the Earth as we have on this planet needed something from outside, or is irresistible for life to pop up as soon as possible on this planet. I would like your comment about that. And the second question is about, somebody has mentioned that we have various living organisms which can stand extreme condition. One which is the most impressive one is the tardigrades, which are very complex living organisms, and they have apparently 1,000 times more resistant to radiation than any other organism on this Earth. Why the hell nature had to develop something which is 1,000 times more resistant to radiation, considering that life has evolved below the atmosphere, so in conditions which are basically protected from radiation in a substantial manner? If anybody has an answer to that, I would really like to hear.
One comment to say about the time to clement conditions after the last giant impact can be much quicker, actually. It could be as little as 10, 20, 30 million years. And so return to clement conditions can be very, very rapid. We expect it probably cycled a number of times.
But I think you also have to take into account what we actually know about the evolution of life on Earth. And the oldest evidence that's been published in the literature, it was claimed that there's one graphite particle in a zircon, which is 4.1 billion years old, which has the right isotopic composition to possibly be biological. But because of the context, we can't distinguish whether it's actually biological versus abiotic. And then when you move forward, it's actually 3.8 billion years that we actually have some sort of reasonable evidence for life, which is essentially a heavily metamorphosed black shale in Greenland that was discovered by Minnick Rosing. He's a Greenland geologist based in Denmark. And then, that's only carbon isotopes again, but you've got the context now, which is a black shale, which, okay, that seems reasonable. And then you move forward to stromatolites at 3.5 billion years old. So there's still, the whole of the Hadean, essentially, which we don't really know about for life. But I guess most people would think probably since you've got photosynthetic life by perhaps 3.8 billion years ago, there's some history in the Hadean, and probably life evolved in the Hadean. But we don't know where, and there's uncertainties of hundreds of millions of years. So to say that life, and some people say life, got going really quickly, what does that phrase mean, really quickly? We just mean that between 4.5 billion years ago and 3.8 billion years ago, it got going. But that's not most people's concept of really quickly.
Then the other part of your question about the timescale for Oumuamua, it's candidate home systems. It would have come from anywhere from 1 to 6 and a bit million years ago, but we don't think those are the likely systems because it was coming too fast. So maybe with further releases of Gaia, they can trace farther back in time. But some have calculated that an interstellar object might pass 20 star systems relatively closely every million years or so. So the timescales aren't that long.
The tardigrades, I think, is the same issue I was talking about. They're adapted for dryness, and it happens to be that that's the same adaptation for radiation. Like Deinococcus, for example, is the same adaptation for dryness and radiation resistance. It's basically very good DNA repair.
Mm-hmm. There's a body of work that actually, looks at this, I don't even know what to call it, synergistic happenstance of being much more resistant to one environmental insult when you're resistant to another environmental insult. And they tend to go together. So extreme cold resistance and radiation resistance and high salt tolerance. And it appears to be that the molecular stabilization that organisms have developed stabilize them against all of those different insults, because all of those impose the breaking of bonds and the destroying of the information content. So it's not really surprising in retrospect that there is this tremendous amount of overlap in these adaptations. And so one of the amusing things is that these organisms presumably didn't adapt to resist high radiation, for example, that they happen to be pre-adapted. But that's not surprising either because, frankly, pre-adaptation by happenstance is a major evolutionary circumstance that leads to, innovation. So I think, yeah, Keith.
Yeah, an observation and a question. There's a lot of discussion as to whether something can come from one planet and hit another and be viable. NASA did that experiment inadvertently in 2003. Space Shuttle Columbia was carrying 6 vials of C. elegans, nematodes, and they were in little vials, and they were in the mid-deck locker, not designed to re-enter the atmosphere. The vehicle was torn apart at Mach 19, 60 miles up. Outside temperatures and the materials were over 1,000 degrees. One of these, the vial landed, it was found, it's opened up. 5 of them had viable C. elegans. They all live in Minnesota now. And a few of them were sent to the space station. So we did that. And to piggyback on Lou's question, first of all, if you have a rock, don't let NASA design it. There are a lot of them outside. But the issue of whether something could come back in, why don't y'all just get some rocks? I mean, rocks, drill holes in them, put things in it, throw them down to Earth from space. I mean, you can pick you can pick Antarctica, pick someplace. Instead of discussing whether or not, why don't you just do it?
Well, funding.
Well, other than that notwithstanding.
People have proposed these sort of things, and they have never been funded.
Pete's head's down.
Right.
Jay's done it.
We had proposed to do that by inoculating shuttle tiles with our genetically labeled organisms so we wouldn't confuse them with background. We were absolutely flatly turned down by the Space Shuttle Board, which is why we had to use sounding rockets. We would have preferred to use some of the leading edges of the shuttle, which are very porous tiles.
But wouldn't this sort of answer a lot of these questions a lot more quickly than just math? And
well, the Biopan experiment did some of that. The Gorno-Hornix experiment, they did re-entry.
Yeah.
Where's my other blue cube?
Hi.
Yeah, hi, Lynn.
Yeah, actually, I think my comment was pretty much covered, basically, that we did not evolve to play the violin. That happens to be come along with having manipulative capabilities the next time you're thinking about it, or much less the bagpipe. But since I had the floor, responding to Ben, we did work years ago looking at photosynthesis just below the surface, which a lot of people have done. So you can go probably even a couple of centimeters below the surface of Mars and still be photosynthetic. You certainly can be protected by sodium chloride or gypsum and still have enough photosynthetically active radiation and block out the UV. So there's sort of ways around it. Now, there are other problems with being the surface, but there are ways around it in terms of the photosynthesis question.
Yeah, and Carl Sagan had a early paper about the euphotic zone where the UV gets attenuated faster.
Interestingly enough, he reviewed my paper, so.
Yeah. But the other thing is those organisms near the surface on Mars will be subjected to all the oxidants in the atmosphere. So it's not clear they would survive on Mars.
Specifically the UV that you can worry about.
Yeah.
Yeah, I just wanted to make the quick point that I think atmospheric entry is the least difficult problem here. I think it's well established that there's only a few millimeters of heat pulse that penetrates into these rocks. And, Jay, as you and Milosh and company pointed out a while ago, a meter-sized body is the right size for shielding you against cosmic rays during interplanetary transfer, and it's also in the sweet spot for being decelerated onto the surface of the Earth. So I think atmospheric entry it's not a non-issue here. It's the easiest part of the problem.
But it gives you a size constraint.
Problems,
Yeah, it gives you a size constraint.
I'm coming back, sorry, here again, Jean-Pierre. I'm coming back to the re-entry process. So there was one experiment which was done, just one, named STONE. And this was done by ESA and Roscosmos through Biopan experiment, but then there were also some organisms, lichens and some bacteria outside of this big device which comes in through the atmosphere, and this the survival failed. So at that time we said, okay, this is not possible with a dense atmosphere like on Earth. But afterwards we more and more thought about what kind of material was used, and this was a kind of granite and eroded granite. it's completely disintegrating. And the same happens also with sedimentary rocks. So it might be different with basaltic rocks or whatever you are using. So there's a lot of potential to do other experiments in future. And I would also suggest to do such kind of experiments in future with re-entry devices.
I'd like to solve this problem democratically. So you've all expressed a range of opinion. I'd like to hear whether you believe that there's a greater than 50% chance that interstellar panspermia took place. And if your answer is no, what do you consider the major weak link in the chain of probabilities to make it happen?
So just to clarify more, interstellar only, and ever somewhere in some galaxy in the universe, or
No,
vis-a-vis Earth?
For us, the Earth.
Coming to us from
Coming
another
to us.
Correct.
Okay, that's
Correct.
considerably
Correct.
narrower. And who thinks it's more than 50%?
And we all have to vote, the whole audience, not just us?
Yeah.
No, you're on the spot. It's up there.
Everyone's voting.
All right.
More than 50% chance. Wow. Look at I trust these people. They're voting yes. Okay.
No.
So do we conclude that no one on the panel believes that this is a likely possibility?
50% is a pretty high bar. Do
Okay,
I think it's-
10%.
Well, I think putting a number
So,
on it is
okay,
kind
if
of
the answer is
goofy.
no, what is the weak
Do
link?
I think it's plausible? Maybe, yes. Probably possible. Does that mean 50%? Not even close?
Yeah. I mean, look, I mean, on this whole question, right? Like, available evidence, data, what we really need to ask a good scientific question is hard.
This is
right
not
now
a
it's
good
very
scientific
speculative.
question. This is based on your prejudice and what you know about your field.
Right. I mean, so I would
an
say,
American vote, Bill.
my own prejudice is I think life is probably indigenous, but I think you can't rule out the other possibilities. It's dangerous to rule out the other possibilities without evidence. So we have to explore these roads and see where they go. And maybe in the end, that leads us to, even if it doesn't lead to the answer that panspermia happens, it leads you to maybe interesting, better questions to ask about how life originated on the Earth, and that's a good rationale.
And so
Well, it's not mutually exclusive either.
Can I
okay, so-
rephrase?
So I'm a scientist, so I base my opinion not on prejudice, but on evidence, and there isn't any evidence that it happens, so I give it 0 at the moment. But if you show me some evidence that is convincing, then I'll change my
But you're
mind.
not averse to people looking for evidence,
Yeah.
right? Yeah.
So the weak link is no evidence. What do you think is the
Can I rephrase this?
Yeah,
Because
go
this is a
ahead,
question that I think
yeah.
about a lot.
Yeah.
We go to Enceladus. We find that there is life in the plume, and it has the identical DNA, has the identical biochemistry that we have on the Earth. Okay, so that could mean two things. It could mean that panspermia seeded both Earth and Enceladus.
Single genesis.
Huh?
Single genesis.
Okay, and then, or it could mean that our biochemistry is the most favored by the universe, and what we found on Enceladus was a second genesis. So how would you guys say we should interpret that probable, or not probable, but a possible result that the biochemistry on Enceladus is identical to ours?
Well, Occam's razor, I guess you have to invoke that. And I think that it's harder to argue that we have a completely optimized biochemistry because so many other variants are possible with it.
So you would say that we couldn't say it was a second genesis?
Yeah, I think we could not.
We could not. Does
Even if
everyone
it
agree with
was
that?
one, I don't think we could distinguish.
Is the probability high enough for panspermia from another system that we would have to conclude we could not say it's second genesis?
Well, no, they could have seeded us or we could have seeded them, right? It doesn't have to come from outside of our star system.
I'm not, okay. Well, I'm not worried about that one. I'm worried about.
You're talking about first genesis or second genesis, but I'm saying that it doesn't have to come from outside the star system.
Okay, so maybe okay, scratch the first, second genesis thing. Just tell me, could we say that it was endemic to our solar system, or did it come from somewhere?
Yeah, I would remember a statement by Norman Pace when asked a similar question just a couple of decades ago. But already then, because of what we knew about genetic sequences, he said he felt that it would be possible to tell whether it was truly the same life as on Earth or different, because of the details of how the catalysis was done by various protein sequences and so forth.
Yeah. Let me just say one other thing too, right? The difficult part about this problem is if you're just looking at probabilities, I can tell you the probability of getting Earth ejected to Enceladus, to Europa, or whatever, is very low, okay? But how much is enough, right? Like if you get just this tiny little bit there, but that little bit happens to have life and it does something, maybe that's enough. And I don't know how to address those questions at the moment. And I'm hoping to hear more at this conference. Maybe people will have ways to address that question.
Okay,
but I think that's the state of the field, right?
It's even lower. Would you agree to this? It's even lower from another stellar system.
Oh, absolutely.
Sure.
So Gary, you're going to answer these things when you stand up here, right? You're saving them up, and you're going to ask them because you know this chemistry. Yeah.
Another interesting point might be, though, that if we do find life on Mars or Enceladus or somewhere, and it is a life that had a common origin with us, it might be possible to get an approximate date for the time, because the molecular clock measuring the mutations that occur on a sort of a roughly steady pace gives a way to date when things happen.
Off of the Earth, too?
Yeah, sure.
Rate of,
Well, you might have to take into account radiations and things like that.
That would be very difficult to actually parse.